This is lecture 2. We're going to start off with a review in terms of the regulation of genome expression. Then we're going to talk about prokaryotic transcriptional regulation. We'll specifically address genetic switches such as the trip operon and the lac operon. Do your readings as per usual. Let's first review genomes. All known life forms possess a genome. It encodes the information to construct and maintain an organism. Most genomes are made of DNA, but some viruses do have RNA genomes. This is a point you have to be a little bit careful on. Viruses are not usually considered living things. Viruses, they're not made up of cells. They don't have cells. They need cells to replicate. So viruses are not really living things. That being said, some viruses have DNA, some viruses have RNA. So we can say some viruses have DNA genomes, some viruses have RNA genomes. The release of the biological information stored in the genome requires genome expression. The first product of genome expression is the transcriptome. And the transcriptome is essentially the repertoire of RNA molecules present in a cell at a particular time. So it's all the RNAs in that particular cell. Now, to give you a snapshot of the transcriptome, what you can do is a DNA microarray. Now, for this course right now, we don't have to worry about how you do a DNA microarray and the technique behind it, but what you do have to do is to be able to read a DNA microarray. So the DNA microarray is giving you a snapshot of all the RNA molecules in a cell at a particular time, and how you read the DNA microarray is just by simply thinking of it as a table. So if it's a table, you know there are rows and columns. The rows are simply our genes. So you can have gene A, gene B, gene C, etc. on and on and on. And obviously this table is really, really small. The columns are essentially our samples. So here are a number of different breast samples, here's a number of different liver samples, here's a number of different ovarian samples, etc. And the color simply tells you how much of that particular RNA is in your particular sample. Red means a lot, green means less, and black is essentially in the middle. So we can look at a particular row as an example. Now again, the table's really small, so over here I've tried to kind of rectangle off one particular row, but obviously there are a couple of different rows in here. So let's say this, for example, is gene D. Well, if you look at gene D, if you go down over here and look at the liver, you can see, oh, in the liver, there's a lot of red. So you can imagine there's a lot of RNA for gene D. There's a lot of D being transcribed in the particular liver. If we look at the breast samples, well, kind of green, kind of black, so it's lower in expression. And then in something like the brain, it's pretty green, so it's even lower in expression. So again, DNA microarray gives you a snapshot of the transcriptome, tells you about the RNAs in the cell at a particular time, and you read it simply like reading a table. Traditional colors, red means more of the RNA, green means less. The transcriptome is maintained by the process of transcription. The second product of genome expression is the proteome. And the proteome is the collection of proteins in a cell. It defines the biochemical functions of the cell. So all the proteins in a particular cell, that's going to be the proteome. Now, just like a DNA microarray gave you a snapshot of the transcriptome, while a 2D gel electrophoresis gives you a snapshot of the proteome. For our course right here, you don't have to worry about how you do a 2D gel electrophoresis, but you should be able to read a 2D gel electrophoresis. For the figure here, there's actually two 2D gel electrophoreses. There's in fact one for the brain and one for the liver, so we can divide it up here. Now remember, a DNA microarray is read like a table, while a 2D gel electrophoresis is read like a graph. So for your standard graph, there's going to be a y-axis and an x-axis. Notice on the y-axis, you have molecular weight going from low to high. And on the x-axis, you have isoelectric point. So that's the pH at which a particular molecule, protein in this case, carries no net electrical charge. And this goes from acidic to basic. And the different splotches here are going to be the different proteins. So for example, if we look at this red splotch here, you can see that's going to be a particular protein. It's going to have a pretty high molecular weight when you look at the y-axis. And for the x-axis, you can see it's pretty acidic. Now, this is the human brain on the left. You have the human liver on the right. So again, you read it the same way. You're going to have your particular axes. Now, your 2D gel electrophoresis graphs are usually not colored in for you, but they're actually colored in for you here. And in fact, what they show here is a comparison between the human brain and the human liver, red being proteins that are similar in these two different things, and blue being proteins that are different. So as an example, you can look at this red splotch over here. That's going to be a particular protein. Again, we said pretty high molecular weight, pretty acidic. Well, that is actually this particular protein in the human liver. So if you compare the brain and the liver, it should be pretty obvious to you then that, well, the brain and the liver, they can have similar proteins. They can also have them in different amounts, right? Look at the liver splotch. It's bigger. The one in the brain is a little bit smaller. So similar protein, different amounts. As well, if you think about the brain and the liver, they're going to have some unique proteins. They're going to have different proteins, like, for example, this blue one here in the brain, maybe around a middle molecular weight, still pretty acidic. But if you look at that same spot in the human liver, you can see that 2D gel electrophoresis doesn't have it. And that is another important point. They can have different proteins. The proteome is maintained by the process of translation. And obviously going from your genome, DNA, to transcriptome, RNA, to the proteome, protein, is also known as the central dogma of molecular biology. The different cell types of a multicellular organism contain the same genome. How do you produce different cell types? If you think about something like a neuron versus a lymphocyte, they're going to look really different. They're going to behave really different. They're going to have really different functions. But from a particular individual, they're going to have the same genome. How do you create these differences? There's going to be differences in genome expression. The human genome only has about 25,000 protein encoding genes. At any one time, only 30 to 60% of the genes are expressed in a particular cell. Expression of almost all genes varies from one cell type to another. And we've actually already seen this, right? Think about our comparisons between the human brain and the human liver. Remember, the red splotches, those were proteins common to both different kinds of cells. Blue were specific proteins. And again, when you compare these two different kinds of cells from a particular individual, they can have some of the same proteins. They can have them in different amounts. And they're going to have unique proteins. They're going to have different proteins. Genome expression is regulated at many steps from DNA to protein. Here we have an elaborated central dogma, and essentially this is an outline of all the different ways we can regulate genome expression. Essentially the things we'll be talking about in the first section of this course. Just to quickly review, remember you have your genome, it's all the DNA in a particular cell. Here's the transcriptome, all the RNA, proteome, all the protein, interactome, those are all the different interactions. Typically, they refer to protein-protein interactions, although there's some variation there. That's the interactome. And then the metabolome, don't forget, in your cell, it's not just DNA, RNA, and proteins. You have all sorts of other things too. You have lipids, you have nutrients, waste. Think about glucose, metabolites, those kinds of things. And all the other stuff, all the small molecule metabolites, that's the metabolome. So if you think about the genome, your DNA can be replicated. It can be organized. You can have heterochromatin. You can have euchromatin. You can have transcription to RNA. You can have post-transcriptional modification. You can have splicing. You can have translation for your proteins. They can be post-translationally regulated. You can sort them. They can have different localization. And all the red lightning bolts here are all the different ways you can control genome expression. You can control what comes out of your DNA. Again, pretty much all the cells in your body have the same DNA, but the cells are going to be different because of genome expression. The first one we'll talk about in detail is going to be the regulation of transcription. Why are we starting with transcriptional regulation? Well, this kind of regulation is crucial for defining cell types, say in multicellular organisms, right? How we get a neuronal cell versus a lymphocyte. Also, this is how you respond to extracellular stimuli. In both multicellular and unicellular organisms, it's transcriptional regulation that plays a key role. There are other ways they can respond to, but definitely transcriptional regulation plays a key role. Recall from your prerequisites that DNA is transcribed into RNA by the enzyme RNA polymerase. Now we're going to quickly review how RNA polymerase works here, but this is just a very quick review. If you didn't learn it, if you don't remember it, if you took a prerequisite and did poorly, it is your responsibility to get caught up. This is not the prerequisite course. This is this course. It's a second-year biology course. So what we would expect you to understand is that this in blue is your RNA polymerase. Here is the DNA double helix. And you can see here the direction of transcription, right? RNA polymerase reading the template from 3' to 5'. Here are your ribonucleoside triphosphates. What's going to happen is they're going to go through this uptake channel. And then at the active site of RNA polymerase, it's going to cut off 2 of the phosphates and add on the next phosphate sugar base. Here is the new RNA transcript. This is the 5' end. So of course you are making your RNA from 5' to 3', right? You're adding on to the 3' end. What happens afterwards is you have a short region of a DNA RNA hybrid, a short helix, and then the hybrid will separate and you have part of your RNA starting to leave. Your DNA double helix will reform. And so of course RNA polymerase will be the key enzyme involved in transcription. Again, recall as in from your prerequisites. So if you don't remember, go back and review prokaryotic transcription. Very quickly here, as a quick review, here is your RNA polymerase core enzyme as in you don't quite have the sigma factor yet. What happens is the sigma factor can combine with the RNA polymerase to make the RNA polymerase holoenzyme. Notice here you have your promoter. And for your prokaryotic promoter, it's this particular region that includes these 2 red sequences. So notice they bracket off for you where the promoter is. It's not anymore before, it's not anymore after. It's these 2 red sequences and the area here in this particular case. May be different for some other prokaryotic promoters. The promoter is the region of DNA that indicates the transcription start site. It may or may not contain the transcription start site, but it indicates it. So here you have your RNA polymerase holoenzyme. So again, core enzyme plus the sigma factor. Notice the sigma factor is what binds to the promoter, binds to the 2 red sequences. And notice how it's positioned the RNA polymerase, right? The RNA polymerase is so positioned so that the active site is way up here. It's in fact a little bit after the promoter, a little bit after this particular red sequence. Now we can zoom up over here so you can see that the RNA polymerase unwinds the particular DNA and then transcription will begin. Notice in light blue you see the RNA. And the transcription has begun, in fact, a little bit after the promoter in this particular case. So in this particular case, the promoter did not contain the transcription start site. The transcription start site was after the promoter, right? The active site was positioned after, so that's where transcription will begin. If we keep on going, you can see that once about 10 nucleotides are synthesized, the sigma factor gets released. That allows for transcription elongation, so the RNA polymerase keeps on going. Eventually what happens is the RNA forms this terminator sequence. You get transcription termination. Again, everything's encoded for by the DNA. It's not something separate. So the RNA is going to form this particular structure after it's transcribed, and then you get transcription termination. So that again is a very quick review of your prokaryotic transcription. Now, what we see in both prokaryotes and eukaryotes is that genes can be transcribed at different efficiencies. If you look at your DNA over here, you can see gene A and gene B. You can see gene A can be transcribed a lot, gene B transcribed a little bit less. Now, definitely not all genes are protein coding, but very often, not always, very often, if it is a protein coding gene, more RNA would mean more protein. There's definitely other ways of controlling genome expression, and that can definitely play an influence, but there does seem to be a trend with more RNA, you get more of the particular protein. So if genes are transcribed at different efficiencies, how does that occur? Gene expression in both prokaryotes and eukaryotes is actually regulated by gene regulatory proteins, specific proteins also called transcription factors. Now, these specific proteins, also called transcription factors, bind specifically to regulatory regions of DNA. These are also called cis elements. Now, for this particular course, I don't want you to get too caught up on cis versus trans. For a discussion of transcriptional regulation, cis elements, just as in specific DNA sequences and they're going to be on the same DNA molecule as the gene. So you have specific proteins, gene regulatory proteins known as transcription factors, binding to specific regions of DNA to really control gene transcription. The gene regulatory proteins, those transcription factors, can turn genes on, in which case they would be positive regulators. They'd be called activators. Also, some of these proteins might turn genes off, in which case we'd call them negative regulators or repressors. And we'll see an example coming up. Let's do a quick review just to make sure we're all on the same page. So remember, genes can be transcribed at different efficiencies. Some genes transcribed a lot, some genes transcribed not so much. Why is this the case? Well, in both prokaryotes and eukaryotes, you have specific proteins called gene regulatory proteins, also known as transcription factors. They bind to specific DNA sequences, and these gene regulatory proteins, some are going to be activators of transcription, some are going to be repressors of transcription. And for this seemingly simple concept, some scientists actually won the Nobel Prize. These gene regulatory proteins were first discovered using bacterial genetics, and these scientists working on E. coli said anything found to be true of E. coli must also be true of elephants. And although the mechanisms are not exactly the same, definitely the concepts are true, definitely the concepts remain the same. And although this concept kind of seems simple to us now, hopefully you can understand the importance of their discovery and why this was awarded the Nobel Prize. This work explains why all the cells of a particular individual, whether it's a human, dog, cat, elephant, can have the same DNA, but the cells can vary widely. It also explains how cells respond to their stimuli. So it is a very important discovery. So we're going to now discuss in more detail bacterial transcriptional regulation. We're going to discuss eukaryotic gene regulation in more detail next class. Eukaryotes will be a little bit more complicated, so let's make sure we understand the basics in our bacteria. For E. coli, it's a great model organism. It's well characterized. It's a unicellular prokaryote. It has one chromosome of circular DNA, encodes about 4,300 proteins. Many are transcriptionally regulated by food availability. So we'll really see how cells respond to stimuli such as food and nutrients. A prokaryotic feature is that multiple genes can be transcribed into a single RNA molecule, what we call an operon. The first example we're going to talk about in detail is the tryptophan operon. Now, importantly, there are other tryptophan operons. For this course, unless you're told otherwise, you should assume we're talking about the E. coli tryptophan operon. Make sure to know this example well. So the tryptophan operon, also what we call the trip operon, encodes for 5 genes, and the genes encode for enzymes that play a role in tryptophan biosynthesis. Transcription is regulated by a single promoter. That's why it's a trip operon. Here is our schematic. You can see this is part of the E. coli chromosome. Definitely not the whole thing, but this is a part of it. You can see there is one promoter here. Here are the 2 yellow sequences. This is what the sigma factor would bind to. And then in the middle here, we have a particular sequence we call the operator. Notice there are 5 genes named for you E, D, C, B, and A. Notice what happens from this one promoter is you make this one long mRNA molecule, and of course you encode 4 enzymes here for tryptophan biosynthesis. There's going to be 5 different proteins encoded for. Notice on the mRNA here, you do start transcription in the promoter in this particular case. Notice there's this light blue area. This would be the 5' untranslated region before the first AUG. As well, importantly for your proteins, each one of these enzymes is essentially necessary for tryptophan biosynthesis. All of these enzymes play a role in making tryptophan. And when scientists typically refer to the trip operon or the tryptophan operon, they may be referring to the particular DNA region, all shown for you here, or they may be referring to the system, and we'll see how this system is regulated. The tryptophan operon, TRP for short, has obviously a promoter, and the promoter can exist in 2 protein-bound states. There's 2 different options for proteins binding to the promoter. The first being the promoter could be bound by RNA polymerase, and obviously if it's bound by RNA polymerase, the trip operon would have gene expression being on. The second option is where the promoter is bound by a tryptophan repressor protein, right? This is going to be a repressor of transcription. So if there's a tryptophan repressor protein, what happens is the trip operon is off. The tryptophan repressor binds a specific DNA sequence of the promoter, what we call an operator. Here's the promoter region of the trip operon up close. You can see the region they bracket off for you being the promoter. Notice the numbering system. Don't forget from your prerequisites that +1, that's going to be the first nucleotide, right, on the DNA, the first one that is transcribed. So whatever this is going to be, maybe this is a C, the first RNA would be a G, etc. Now, you can count forward, right? +2 would be the second nucleotide being transcribed, +3 the third one, up to here +20, the 20th nucleotide being transcribed. You can also count backwards. There's no 0, but 1 before where you start transcription would be -1. 2 before where you start transcription would be -2. And you can count all the way back here. You can see to the -10 and -35 regions. That's where the sigma factor will bind. And a little bit in between them, you'll have the particular operator. So if the tryptophan repressor binds to this operator region, well, RNA polymerase then cannot bind, and the trip operon would be off. So tryptophan repressor protein binding to the promoter is going to block the promoter access, then RNA polymerase cannot bind. That's going to negatively regulate the trip expression, but the tryptophan repressor DNA binding activity is regulated. You can't always have the repressor protein on the DNA, or else you'll never get transcription. What happens is the tryptophan repressor protein must bind 2 molecules of tryptophan to then bind to DNA. The repressor and operator provide a simple switch to control tryptophan biosynthesis according to the availability of free tryptophan. Here's an overall picture which I think really helps. So remember, we are talking about the tryptophan operon. Unless we're told otherwise, we're thinking the E. coli tryptophan operon. Here's your basic setup. So this is the promoter region. We know downstream here there's going to be 5 genes. If you have those 5 genes being transcribed, you're going to translate those into proteins, and those 5 proteins are going to help the E. coli make tryptophan. Here's the promoter region. You can see the start site of transcription in here. You can see -10, -35 regions where the sigma factor binds to. You can see the operator where the tryptophan repressor might bind to. Now think about your general picture here. Let's say in the first case, the E. coli is experiencing low tryptophan levels. Well, on a very general level, if the E. coli has low tryptophan levels, you can think, hey, I better transcribe these genes, right? The E. coli better transcribe this stuff. It better get those proteins. It better then make its own tryptophan. And that's exactly what happens, right? Under low tryptophan levels, the repressor is inactive. RNA polymerase can bind. It can transcribe this. You transcribe the operon. You get the RNA. You translate. You get the 5 proteins that help it make tryptophan. Now think about what happens when there are high tryptophan levels. If the E. coli already has high tryptophan levels, well, it wouldn't need to make more tryptophan. It wouldn't need to transcribe the operon. You wouldn't need the 5 particular proteins to make more tryptophan. Tryptophan's already high. So what happens with high tryptophan levels is tryptophan binds to the repressor is then activated, the repressor goes to the operator, and if the activated repressor is on the operator, well, it stops RNA polymerase from binding. It blocks RNA polymerase. So the genes are off. Now, if you think about this figure for a sec, though, hopefully you might notice that there's a little bit of a problem here in that, remember, to activate your repressor protein in this particular case, you actually need 2 molecules of tryptophan to bind to it. They just show you 1 molecule binding to it. So hopefully the trip operon makes sense to you. If not, make sure to rewatch that little section. Definitely as well, always consult the textbook. Now, if we look at the structure of the tryptophan repressor protein, we actually see that it contains what we call a helix-turn-helix DNA binding motif. A helix-turn-helix DNA binding motif is actually a structural pattern. It's a shape pattern. And this structural pattern we see in many different proteins, not all, but many different proteins. And in fact, it's the most common DNA binding motif, as in for many proteins that bind to DNA, they have this particular structure. Here's what the structure essentially looks like. So it's essentially a helix, then it turns, then it's a helix. And for many proteins that have this particular shape, what they can do is they can bind to DNA. And in fact, they bind to the major groove of the DNA helix. So if we look at the tryptophan repressor protein, this is what it looks like. Notice there's a lot of different shapes in here. There's a lot of different things. But one of the things it does have is a helix turn helix. And it's actually over here. So helix, as in this tube, turns, and then another helix. And you can kind of see it over here too. There's a helix, turns, and there's another helix. What happens is if the tryptophan repressor binds to 2 molecules of tryptophan, right, shown for you in red here, what happens is you see the helix turn helix here now is in the right shape, it's in the right conformation to bind to the major groove of DNA. And that's exactly what it says down here. Tryptophan binding induces a conformational change in the protein. Now it has that right shape to fit in the major groove. The second example we want to talk about in detail is the lac operon. And again, just like the trip operon, there's other kinds of lac operons, but for this course, unless you're told otherwise, you should assume that we're talking about the E. coli lac operon. And this is an example you should know in detail. The lac operon encodes for 3 genes required for the transport of lactose into the cell and for its catabolism. So moving lactose into the cell and catabolism, as in breaking it down, using it for energy. The lac operon enables the use of lactose in the absence of glucose. Now, for this point, there are 3 extremely important points, and this will help you to understand the lac operon. So make sure you note this down. Number 1, E. coli's first choice is to use glucose, right? E. coli wants to use glucose. Number 2, when there is low glucose and high lactose, it will then use lactose. Both of those conditions, though, must be true to use lactose, right? You need low glucose and high lactose. Both of those conditions have to be true. Number 3, the lac operon is what is turned on when it wants to use lactose. So keep in mind those 3 rules, and that should really help you explain when the lac operon is on. So for example, if glucose is high and lactose is high, well, you're not using the lac operon. It's just going to use glucose. If glucose is low and lactose is low, again, you're not using the lac operon. It's going to use something else. It's got to be low glucose and high lactose. Both of those conditions have to be true. Then it's going to use lactose. Then it's going to turn on the lac operon. That's how it works at a general level, but we need to also understand the molecular biology. For the molecular biology part, there is dual regulation. There's both positive and negative control. There's in fact an activator as well as a repressor. What the lac operon needs for transcription is an activator protein to help activate transcription, and that's specifically CAP, the catabolite activator protein. And it promotes lac expression during low glucose and high lactose conditions. And there's also a repressor protein, right? So you need to remove the repressor protein if you want transcription. And the repressor protein is the lac repressor protein. It inhibits lac expression when there is low lactose. Now we're going to go over how it works, but make sure to look at the textbook readings for this section, including the figure and the figure legend. Definitely take a look at it. Let's look at our general setup for the lac operon. Here is part of the E. coli chromosome. They don't show you the whole lac operon, but a little bit of it here. You can see over here, this is the CAP binding site where the particular CAP protein binds, the activator binds. Here is the RNA polymerase binding site, the promoter. The promoter essentially starts at this bracket section here and then ends over here. You can see in yellow, this is where the RNA polymerase binds, as in where the sigma factor binds. In green, this is the operator, and the operator is where the lac repressor protein binds. Notice the start site of RNA synthesis is actually in this particular promoter. You start transcription over here, and then eventually you get over here. This part's going to code for LAC-Z. LAC-Z is the first gene of the lac operon. It encodes beta-galactosidase. It breaks down lactose to glucose and galactose. Also, it can convert lactose into allolactose. Now, the first condition we want to talk about is what happens with glucose and no lactose. I'll also mention something here is that a lot of students will ask, okay, here it says yes glucose and no lactose. And before we kind of mentioned things like high and low, well, how high is high? How low is low? What about medium high, medium low? I'll say those kind of intermediate levels, that's a little bit beyond this particular course. In later courses, you'll be able to calculate, oh, with 30 nanomolars of glucose and 20 nanomolars of lactose, what's the probability of gene expression? But that's a little bit beyond here. So for this course, yes glucose as in high glucose, no lactose as in low lactose. So already, hopefully from our rules, you know that if glucose is high and there is no lactose, well, E. coli is just going to use glucose, right? That's its first choice anyways. So definitely the lac operon will be off, but we got to understand what happens at the molecular level. When lactose levels are low, the lac repressor is bound to the operator. So if the repressor's there, you're not going to have RNA polymerase binding. Lac operon gene expression will be off. As we'll talk about, it's never quite completely off. Increased lactose will remove the repressor from the operator. Increases in lactose increase levels of allolactose, and allolactose is related to lactose. What happens is this conversion requires beta-galactosidase. As in, if lactose is low, well then beta-galactosidase doesn't convert as much into allolactose. But if lactose is high, well, beta-galactosidase will convert more of it to allolactose. So if allolactose is high, notice it's not quite lactose. If allolactose is high, well, allolactose will bind to the lac repressor. And if allolactose binds to the lac repressor, it'll cause a conformational change in the protein. It's going to decrease the DNA binding activity of the lac repressor, and it's going to release the repressor from the operator. And this is essentially what we're going to see. So as we said, with low lactose and high glucose, the lac repressor will be on there. But if you increase lactose, right, if you increase lactose up here, well, what happens is with increased lactose, beta-galactosidase will convert some of it to allolactose. And if allolactose is high, well, then allolactose binds to the lac repressor, causes a conformational change, decreases the DNA binding activity of the lac repressor, releases the lac repressor from the operator, and you can see in this case now, the lac repressor is gone. So if you think about it, the lactose concentration here really makes a difference, but if you get down into more detail, it really is the related allolactose concentration, right? That really makes a huge difference. Now, notice in this top case, yes, lactose is high, which means allolactose is high, which means the lac repressor is gone, but the operon is actually still off. Why is the operon still off? That's because the glucose concentration also plays a role, and the particular cap protein, the activator protein, is not there yet. Why do you need an activator protein? Well, RNA polymerase binding is actually inefficient to the lac promoter. Efficient RNA polymerase binding to the lac promoter requires cap to be bound. Notice not all systems are like this. For the trip operon, you didn't need an activator protein. As long as the trip repressor was gone, you could have transcription. That promoter was pretty good, had great sequences, had efficient binding to RNA polymerase. In this case, though, the promoter sequences are slightly different, and they don't bind as well to RNA polymerase. So in this case, you need an activator to really help RNA polymerase bind. Cap contains a helix-turn-helix DNA binding domain. So if we are in this particular condition, with lactose, with glucose, we know the operon is off, we know the repressor is off. How can you get cap to bind? Cap DNA binding activity is activated by low glucose. With lower glucose, this activator will bind, it'll help RNA polymerase bind. How does it work at a molecular level? Well, decreasing glucose levels increase the levels of a signaling molecule called cyclic AMP. Essentially what we have is an inverse relationship. If glucose is low, cyclic AMP levels are high, and if glucose is high, cyclic AMP levels are low. We'll talk more about the enzymes involved in this mediation later on, but for now, do remember it's an inverse relationship. If glucose is low and cyclic AMP is high, cyclic AMP binds to the cap protein, causes a conformational change in the cap protein, increases the cap protein's DNA binding activity, and the cap protein will now bind to the cap binding site. And cap's an activator, so it's going to recruit RNA polymerase to the LAC promoter. Let's look at our overall picture for the LAC operon when it's on. So remember, this is the LAC operon. Unless you're told otherwise, you're thinking this is the E. coli LAC operon. And down here, you will have some genes for the E. coli to take in lactose and break it down for energy. Now you can see the conditions here. Low glucose, high lactose. Both of our conditions are met, so we know that the operon is going to be on. However, we must understand what's happening at a molecular biology level. So let's consider both of these conditions. First, we can consider lactose. Lactose is high. That means beta-galactosidase is going to convert some of it to allolactose. If allolactose is high, allolactose is going to bind to the repressor. The repressor then cannot bind to DNA. And that might facilitate RNA polymerase binding. There's nothing blocking the promoter. We must also consider the other case, glucose. If glucose is low, we know it's an inverse relationship, so cyclic AMP is high. Cyclic AMP can bind to cap. Cap can bind to DNA, and cap's an activator. It's going to help RNA polymerase bind. Remember, both of these conditions have to be true. Only then will the operon be on. Only then will RNA polymerase really bind and get transcription here. Now, before we talk about one important complication, let me just say, make sure you review this section, make sure you know it well, because if you don't know it well, you're not going to understand this complication. So pause the video here if you need to, replay the last section, read the textbook here if you need to. With that being said, if we flip back a couple of slides, if you remember when we were talking about the LAC repressor and the LAC operon gene expression, if you recall this slide, we said that when lactose levels are low, the LAC repressor is bound to the operator. The LAC operon gene expression is off, but never quite 100%. As in, if you imagine a condition with low lactose, as in low allolactose, well, the repressor here is binding it, it's really shutting down gene expression. It's off, but never 100%. There's actually a little bit of leaky expression. Why is that the case? If you recall this slide, increases in lactose increase levels of allolactose, which is related to lactose, and that requires beta-galactosidase. So if you increase levels of lactose, what you need is beta-galactosidase to convert it to allolactose. And it's allolactose that binds to the LAC repressor and causes the LAC repressor to be removed from the operator. Now, beta-galactosidase mediates this conversion, but beta-galactosidase is actually the first gene that's transcribed from the LAC operon. So you actually need a bit of leaky expression. Just in case lactose were ever to increase, you need that beta-galactosidase to convert it to allolactose. Another way to think about it is down here. Let's say lactose is low, which means allolactose is low, so we know the LAC repressor is on here. Well, you're not getting gene expression, but you still need just a bit of beta-galactosidase being made, just in case lactose were ever to increase, right? Because if lactose were ever to increase, well, you need to convert it to allolactose, and then you need to remove the LAC repressor. And that's really quite an interesting complication, right? You need just a bit of leaky expression, just to have a bit of beta-galactosidase around, just in case lactose levels were ever to change. Then you could convert it to allolactose and then remove the LAC repressor. With that being said, make sure you review, read the text as well. That little complication's in the text too.